Gender inheritance. Okay, so we have to differentiate as well the structure and the functions of an X chromosome. Since an X chromosome is structurally larger chromosome, it actually carries an assortment of genes. So therefore, usually whenever it's going to be inherited by an individual, the parent having or giving this X chromosome actually gives the trait. Okay, so in the case of Y chromosome, since it's shorter by structure, it only carries male determining factors or male determining genes. Meaning, we would know that an individual would only be male if the sperm cell is carrying Y chromosome. Okay, so any male determining genes are exclusive only for Y chromosome. So any chromosomes that are being carried on the sex chromosomes, for example, if let's just say color blindness trait, it is usually being carried by a female, but males are usually being affected. Okay, so in the inheritance of a specific trait, we have to use Punnett grid as well. Okay, inheritance of sex-linked traits. So we can show them using Punnett grids as well. Okay, so last but not the least, okay, we have to reiterate as well about sex linkage. Okay, so in the case of sex linkage, it actually discusses what type of individual or what gender usually carries a specific disease or a specific trait, or what type of individuals that are being affected by this trait. Okay, so that's about it in theoretical genetics. This is about genetic engineering and biotechnology. So the things that are needed to be discussed under genetic engineering and biotechnology are actually mainly methods and steps on how we are able to alter the DNA. Okay, so since the DNA contains a universal code, we can actually transfer DNA fragments or DNA genes from one organism to another organism that are unrelated to each other, because the DNA contains genetic codes that is universal for each and every type of organisms. Okay, so in the case of genetic engineering, okay, so we are actually able to discuss about recombinant DNA. So in the case of recombinant DNA, this is a type of DNA that has been genetically altered already. Okay, so let's just say if it's going to be genetically altered, so maybe we are going to insert an insulin producing gene, okay, coming from a human to the DNA of a bacterium. So in that way, the process is already producing recombinant DNA. So we just need to elaborate on the steps that is actually required for the DNA transfer. So we have to include as well that we have to discuss as well that genetic code is universal. So just like what I've said a while ago, that since the sequences of bases is common for all types of organisms, whether it's going to be a prokaryotic organism or a eukaryotic organism, it's common for all. So we are able to insert or transfer a specific gene that is being produced by a human to a bacterium, just like what our examples a while ago is. So Now, in the process of genetic engineering, we have to include the methods that are being used. Okay? And of course, we have to explain what the processes are, okay, and their goals. Okay, so for example, we have the PCR, or the polymerase chain reaction. The goal of the PCR is to amplify minute quantities of DNA. So this is actually being used in paternity tests, or especially in crime scene investigations, wherein we are only able to acquire a very, very small amount of DNA samples. So in this way, PCR, we are able to amplify, or to make more copies or replicates of the original DNA sample. Okay, so we have to reiterate. We have to explain further on the process as well. Okay, so next process that are being used, we have to explain about gel electrophoresis. Gel electrophoresis is a process wherein we want to separate organic molecules, or organic charged molecules, wherein whenever we subject DNA fragments into an electrophoresis, or we allow electric current to flow through these DNA fragments, they are going to separate. Okay, the fragments are going to separate. So in this way, we are able to see whether there are like similarities between the samples that we are comparing, similarities and differences on the DNA samples that we are comparing with each other. So let's just say in the case of paternity tests, we would see that if the bands that are being used in electrophoresis, the bands that are very, very similar to each other, it means that these two individuals, or the DNA fragments between these two individuals, are related to each other. So in the case of crime scene investigation, we would see that we would actually solve who the real culprit is if we are able to see like most common DNA fragments between the victim and the suspect. Okay. We are also able to use methods under gene transfer, okay, wherein we're able to cut DNA using restriction enzymes. Using restriction. So in the case of restriction enzymes, there are several types of restriction enzymes, and these enzymes cut the DNA strands at a specific point, producing blunt ends or sticky ends. Since we are going to use it in a gene transfer, then we should be able to use like restriction enzymes that would be able to produce sticky ends, so that we can actually insert the required gene to a plasmid, or to the DNA that is being produced by a bacterium. Okay? So the next thing that is needed to be discussed under genetic engineering is about DNA profiling. Okay, the above mentioned methods, we can actually use these methods under DNA profiling, because we are just actually making a fingerprint of the DNA. Okay, so basically we are actually identifying a person or an organism, because each and every DNA sample is very unique. So there's no two individuals will have the same DNA. So, okay, by using DNA profiling and comparing them with each other, we would be able to see, like, depending on what goal that we are trying to use this DNA. Then, for example, if we are going to use this in paternity tests, we would be able to see who the real father is. So, or who the real parent is. Okay, so under DNA profiling, okay, we have to highlight the use of satellite DNA. Okay, satellite DNA, these are the intervening lengths of the DNA that does not code for a specific protein. So they're just like an excess of the DNA. Okay, so we also need to mention about restriction enzymes. Okay, just like what I have mentioned a while ago, restriction enzymes. Of course, their uses. Okay, so uses such as for paternity tests or crime scene investigation. Okay, so we also need to discuss about the techniques being used in gene transfer. So just like basic steps that are being used in gene transfer. So we should discuss about what enzymes that are being used to cut up the DNA or to cut up the required DNA that we want to insert to another DNA of an organism. We have to include what plasmids are. Plasmids are basically vectors or devices that are being used in gene transfer. So things like that. Okay, so we should be able to use like or give examples of crops or animals that has undergone gene transfer. So for example, the production of BT maize. So BT maize it is actually a crop or maize crop that has actually accept or received a gene coming from a bacterium. Okay. Okay, so whenever when this crop received this gene coming from a bacterium, it is able to like withstand being eaten by, or it is able to actually survive or not going to be eaten by a specific insect. Okay, so in that case, it actually increases crop yield. Okay. So in this case, so right after explain the techniques, we have to explain the benefits and harms of gene transfer as well, because there are also ethical standards that are being debated by several groups of people, wherein they're not really comfortable in using gene transfer as a mode of increasing crop yield or increasing the use of animals as in livestock. Okay, so lastly, okay, the last process that is needed to be discussed under genetic engineering is about cloning. So in the case of cloning, this is when we are trying to produce a genetically identical individual that has been derived from a single parent cell. So of course, the most basic things that are needed to be discussed here is, of course, the process or the method of cloning. And one common example of it is whenever, when Dolly Dolly the sheep was able to be cloned from just one parent. Okay, so we also need to discuss about the advantages and disadvantages of cloning. So there are also like ethical issues that arises from this topic. So these are the most common things that are needed to be discussed under genetic engineering and biotechnology.